Okay, I'm unmuted whenever we're ready to go. Okay, Good afternoon. Welcome to everyone. We welcome you today to another installment of our series on COVID vaccines. We are very pleased to have with us today Dr. Karen Kotloff. Dr. Kotloff is a professor of pediatrics, medicine, and epidemiology and public health at the University of Maryland School of Medicine. She's principal investigator of the NIH-funded Vaccine Treatment and Evaluation Unit at University of Maryland School of Medicine, and associate director for clinical research at the university's Center for Vaccine Development and Global Health. She leads clinical trials to evaluate vaccines against a wide range of infections in the US and in developing countries, including 2 large ongoing trials of COVID vaccines. Dr. Kotloff also has multiple studies in Africa to understand and prevent child mortality. She's published over 250 manuscripts in peer-reviewed journals. I might also answer, Dr. Kotloff and I worked together for many years at University of Maryland, and I know that the Center for Vaccine Development there has been a major player in terms of getting COVID vaccines together and doing the initial testing. And so she was gracious enough to agree to talk with us today about the COVID some of the COVID vaccines and some of the work that they've been doing at the Center for Vaccine Development at University of Maryland. So, Karen, I'm going to hand it over to you. And again, welcome and thanks very much for participating in this series.
Thank you, Glenn. Is my screen showing? I can't see that it's sharing.
Yes, ma'am, you are live. Go ahead and reshare your PowerPoint if you could, please.
Okay.
It was sharing there for a second.
Okay, I hope this is the right version because I didn't see the title, but we'll give it a whirl. Well, thanks so much for the introduction, Glenn, and it's a pleasure to be here, in at least virtually. So I'm going to give an update today on COVID-19 vaccine development from mostly a clinical perspective, with really most of my emphasis will be on messenger RNA vaccines. So first of all, my disclosures. I am site principal investigator of the Moderna phase 3 COVID vaccine trial at University of Maryland and get NIH salary support for that. And am the protocol co-chair for CoVPN's Novavax phase 3 vaccine trial for COVID. And again, that's NIH salary support. So today I'm going to begin by giving you just a review. It's hard not to do this each time, of the rationale for SARS-CoV-2 vaccine. And then I'll talk about the process for vaccine development and the considerations that have gone into choosing spike protein as the antigen and messenger RNA as the platform. And finally, then I'll review the messenger RNA vaccines that are in clinical development as part of Operation Warp Speed, the overall approach, the platforms and their attributes, and the results. Really, I'll focus on the phase 3 trials and an overview of the EUA plans and future directions. And I took a look at Ira Longini's talk, so I tried to not repeat the things that he focused on. So just to give you a reminder, in case you forgot, that we're in the middle of a pandemic that has caused cumulatively in the world 71 million plus cases and 1.6 million deaths. And we're in the middle of a surge. So if you look at these graphs, I hope you can see my cursor, you can see that the number of daily cases since March has gone up, as has the number of daily deaths. And in the United States, we've had a total of about 16 million plus cases and 200,000 deaths. And we're also, if we had a first wave and a second wave, we're now in the middle of a third wave. These are our daily trends in cases, and our cases, deaths, and hospitalizations continue to increase, so that we are now at a rate of more than 1 death per minute. And this is the hotspots in the United States now, which is quite different from previous waves. So it's important when you're considering vaccine development to understand which groups are most important to target. And those that have the greatest need in the instance of COVID are conditions that increase the severity of illness. And so we know that age is an important factor, and you can see compared to young adults that with increasing age, you have a marked increased risk of hospitalization and death. If you have these underlying conditions, they have been pretty clearly associated with an increased severity of disease. And then there are certain ethnic and racial groups that have increased risk, and this is cases, hospitalization, and death. So these are Native Americans and Alaskans, and Asians, Black or African American, and then Hispanics are separated out here. And so you can see that each of these groups have an increased risk. So, but it's not felt that just vaccinating these groups is a good strategy. We really need, as Ira said, to produce widespread immunity, perhaps with the vaccines looking as efficacious as they do, in the range of 50 to 60% of the population, to really get herd immunity and interrupt transmission, and that will take a while. So I understand that there is a range of expertise and knowledge in the people who I'm speaking to, who unfortunately I can't see. And so I just wanted to give a little bit of background about how vaccines are developed. So the first thing that happens when a vaccine is being developed is there are studies in the laboratory which are really designed to look and see as best as possible whether the vaccine candidate is safe and efficacious. And all of those data are presented to the FDA, and you apply for an investigational new drug application. And if they allow you, you can proceed to human trials. Phase 1 trials are always in healthy adults and small numbers of adults. They're first in humans, so safety is your primary concern. And then you're also looking to see whether the immune responses that you think would correlate with protective immunity are elicited by vaccination. And if your endpoints for those studies are met, then you proceed to phase 2. And phase 2, you go from healthy adults to more toward the target population. So they may not be perfectly healthy. You would, in the instance of COVID vaccines, you'd start with young adults and then you'd proceed to elderly because you're going to want to vaccinate elderly. So that's an example of how you get closer to the target population. You use larger numbers, and of course safety is your primary concern. But here you can look at things like schedule and dose response to pick your best regimen for vaccination, how many doses you're going to need and how far apart they should be. And then there's phase 3. And in phase 3, that's your target population. That is who you're going to want to deliver the vaccine to once it's licensed. And always safety is a primary concern, but really the major question that you're asking is when you vaccinate people who are exposed naturally to the infection, does the vaccine prevent disease? Or does it lessen severity? And in the instance of the COVID vaccines, and I'll go over that in more detail, preventing disease was the primary endpoint in most of the trials. So vaccine development is normally a lengthy, risky, and expensive process. And if you look at this top part of the diagram, you can see, I mean, it's very regimented. We do it the same way every time. We go through all the phases. And, you know, you start with manufacturing in small scale, and then you have to be able to scale up to do, you know, large numbers of vaccines. And that's not easy. It's the difference between making a pie for your nuclear family and inviting 100 guests over and trying to make 50 apple pies. You may not have the same quality control. And so that can be challenging, and it's usually not done completely at risk. So manufacturers like to get some idea if the vaccine is going to work before they build the manufacturing capacity to scale. And this whole process tends to take 15 to 20 years. Well, we've done the same thing for COVID in about 12 to 18 months. So how have we done that? We've made these progress charts more vertical than horizontal. And so whenever it's possible to kind of telescope things, so you do initial studies to get an answer. And at the same time, you might be finishing up some of the work in the laboratory. And so you won't go to the next level before you meet that milestone in the laboratory, but you can really do kind of a checkerboard where one milestone leads to your ability to progress further in clinical trials. And the other really important part is that instead of waiting to see what the phase 1 through 3 trials show and then starting manufacturing, our government poured a lot of money into manufacturing at risk. And that's really, you know, if you think of taking this and really putting it where it belongs, you would get much further out. And so that is another thing that helped. And then a third thing, which I'll talk about in more detail, is that we've really, after being almost stuck for a number of years, really advanced the technology for making vaccines and learn new strategies that have shown to be successful for COVID vaccines. And that also has really helped to speed things up. So this is the pathogen. This is COVID, and it's a single-stranded positive RNA with 4 major structural proteins. The one that really stands out are these pink, what we call spikes. And those spikes contain what we call a receptor binding domain. And that is what the virus uses to attach to our cells. And once it attaches, it fuses and then it enters our cells. And if it can't do that, then it doesn't bother us at all. And so it really has an Achilles heel on its surface, but it didn't make it entirely easy to to attack that and use that as a vaccine. And so there were a lot of other discoveries that allowed that to happen. And then there are also these other proteins. There's a membrane protein. Here's the membrane and membrane envelope and a nucleocapsid protein. Those proteins have importance for some diagnostic reasons, but they're not really being targeted per se aggressively for vaccine development purposes. It's really the spike protein because we know that you can prevent infection. And these are really beautiful 3D structural pictures. And what you can see here, so this is the coronavirus, and this is a 3D picture of the spike protein. And this is our cells. And on the surface of many of our cells throughout our body is what we call an ACE receptor. And it is that ACE receptor that's the major target. So the spike protein attaches to that on the cells in our body. And those ACE receptors are quite prevalent in our respiratory tract, both upper and lower. So here is the spike protein attaching to the human ACE receptor. Sorry, I have a kind of jittery computer and it kind of goes on its own sometimes. And then this is what we expect vaccines to do. So these little 3-pronged things are actually antibodies. And what's happening is that those antibodies are coating the spike protein and they're in the way, they're hindering its ability to attach to the ACE2 receptor. And so you can't have infection. And that is the whole goal of vaccination, is to create that interference. They're the antibodies. Okay, so the people who really advanced our ability to develop SARS vaccines were people who were studying respiratory viruses and vaccines too, against respiratory viruses. And so they had learned some lessons. One is that during the 1960s, a wholesale inactivated RSV vaccine, so they basically just killed RSV with formalin and gave it in clinical trials to infants and young children. And they found that when those children 1 year later were exposed to RSV, they had enhanced disease, and they actually, there were deaths that occurred. And that just, really terrified people. And they went back into the laboratory and tried to understand what the problem was. And so there were some theories, and they made these fusion protein subunit vaccines. And that F protein is what the RSV virus, or RSV, uses to attach to respiratory cells. And so they just used that protein, and they found that it didn't work particularly well. And then there was a lot of progress in technologies that allowed, it was structural biology, it allowed scientists to have a look at the atomic level of the structure of the attaching moieties and to find the epitopes on them that provide neutralization sensitive, they're neutralization sensitive epitopes. That is, that when you have antibodies directed at those specific places on the protein, that protein cannot enter the cell. So you have to neutralize the virus, and they learned some lessons. So here we go. We're back now to the SARS-CoV-2 or the COVID virus, and you can see the spike protein on its surface, and corona means crown or circle of light. And this was really an important paper, and Jason McClellan and Barney Graham have really been leaders in the field to understand what about the structure of spike protein is important in vaccine development and what lessons they could learn from RSV. And so they did the 3D structure of the spike protein, and they saw that it had 3 subunits. There's an S1 and an S2, and I guess the other one, I'm not sure. I think the other one must be S3. So it has these 3 components to it, and the S1 unit is the one that binds to the ACE receptor, and that's the first step of infection. And then after that, S2, the S2 subunit engages the cell with a fusion peptide, and it attaches to our cell's membranes. And in the process of that, the whole spike protein undergoes really significant structural rearrangement to fuse with the viral membrane with the host cell membrane. And then the S1 subunit is discarded. So that's the process of attachment. And what was actually also observed through these studies, here is the spike protein before it fuses with the cell membrane. And what you see at its apex these are where all the neutralization epitopes are. So this is what the antibodies have to bind to prevent infection. And then what you can also see, this is the the spike protein. Once it fuses to the cell membrane, you can't see those epitopes anymore. And so it started becoming clear that the proteins that were in the original RSV vaccine that didn't work did not take this into consideration, and they were the proteins were in the wrong structural formation, and antibodies would bind to them, but they wouldn't neutralize the infection. They wouldn't prevent the infection. And so it actually crippled the immune response and made the infection worse. And so the other thing that's important to know is that the pre-fusion form has a hinge on it, and it flaps, and I guess you can see me. So what that hinge does is it hides that red spot, and it goes like this until it attaches to the receptor. And so what you need to do to make the spike protein vulnerable is to stabilize it in the hinge open formation. And so there was really a lot of granularity that you needed to consider to make a spike protein that is likely to be an effective vaccine. And when that was done, this is the Science article that showed in a human trial. Actually, this is with RSV. This was the first proof of that concept, that you actually markedly increase neutralizing antibodies. So it was very effective. This is where I've already told you about those hinge-like movements. And to stabilize the pre-fusion spike protein in the conformation that reveals the neutralizing epitopes, there had to be 2 proline mutations. And that's also been shown to be an effective approach for other SARS viruses. and that resulted in the hinge staying open, and you had more immunogenic and protective spike protein antigens compared to wild type, what you find in nature. So they kind of tricked the virus, tricked the spike protein, and really found its Achilles heel. So this became the approach to developing SARS vaccines, and the spike protein in this conformation is really the central antigen of all the different approaches that are at least being tried by the Operation Warp Speed. And I'll explain a little bit what that is by that program. And the other part that is important to consider is that you can still do more by picking delivery platforms that target the immune responses that are desirable, and they are generally CD8 T cells and CD4 T helper cells, so you get both cytotoxic immunity and antibody formation. And so you want to be able to have a Th1 driven immune response in order to avoid getting enhancement of disease. And the platform that you use to deliver the whatever your, especially so if we're talking about mRNA vaccines, the mRNA can strengthen these properties, and that was another consideration that was really important. So let's move away now from the whole rationale of how the vaccines were developed, and let's talk about how vaccines are tested in this country. So the program that was funded by the Trump administration to develop COVID vaccines is called Operation Warp Speed. It is a name that makes some people nervous, and it's why I was emphasizing some of the points that I emphasized, that that doesn't mean that we're bending and skipping all the steps that we normally go through to develop vaccines. We're just doing some things related to manufacture at risk, and we are telescoping steps without skipping any steps. So Operation Warp Speed, I think the funding that it got from the US government was somewhere around $10 billion, and it's a public-private partnership for strategy and resource allocation for vaccine development. And there is another organization that is an NIH-based organization called Active, and they are also public-private and provide harmonization and strategic advice to this whole enterprise. Then BARDA, part of the US government, is responsible for funding the strategic plan that Operation Warp Speed has developed. And then what happens as a result of this process, what Operation Warp Speed did is they chose, right now it's 3 platforms. They said, we have to have a lot of kicks at the goal in order to make a goal. This is all new. We don't know what works. So we want to try 3 different platforms for making vaccines and have 2 different formulations of each. And so they selected companies that they felt were able to produce in this period of time heavily funded vaccine development and manufacture. And then, you know, the FDA obviously is every part of the step in evaluating preclinical and clinical data and making approvals. And so here are the companies, and they need, it's the company, they're the sponsors for doing the clinical trials. And so who does the clinical trials? Well, There is a consortium called the COVID-19 Prevention Network, or COVIPN, and that consists of vaccine treatment and evaluation units and HIV prevention and treatment units. So these are long-funded vaccine trial sites by NIH, and that's one part. And then there are also commercial research organizations that the sponsor contracts. And in general, there are about 100 sites for each of the trials. And so it's at that level that the trials are performed. And so I talked about this. The FDA has published guidance in terms of what they expect for the endpoints of vaccine. And they wanted the primary endpoint to be prevention of COVID disease. And the secondary endpoint that was prevention of severe disease. And then there are many other endpoints that are part of these trials. And they were being very guarded. And, you know, at that point, when everything was unknown, they said that the minimum efficacy, the point estimate, needed to be no worse than 50%. 50% is kind of low, but you may be able, if you can get high enough coverage, to stop the pandemic, and that the lower bound estimate around that needed to be at least 30%. They made it very clear that people who are at high risk for disease needed to be well represented, and so that includes older age groups, people with chronic conditions, and minorities. And these are double-blind, placebo-controlled trials for the most part. Some have an allocation of 1-to-1, some give 2-to-1 vaccine to placebo. And at that point in time, they said, look, you know, we can't just do these trials really quickly and then walk away. We need to closely follow up these participants for 2 years, which is probably, you know, for reasons that it may already be clear to you, is probably not going to be feasible, but there are other approaches to try to compensate. And then during the trials, there are multiple blood draws. For example, there will be those blood draws, and the anti-NP antibodies will be used to detect asymptomatic infection and to see whether vaccines prevent asymptomatic infection. And those blood draws will be used for a very intensive effort to look at correlates of protection, because it's felt that at some point we're not going to be able to keep doing placebo-controlled studies. And so if you knew what a correlate of protection was, you could do subsequent studies using the endpoint as that immune response, as that correlate. And so that's an ongoing process. So these are the 6 pairs of vaccines that have been put into clinical trial. They've all almost started. So Moderna and Pfizer are the farthest along. They're messenger RNA vaccines. And both are given as 2 doses, a little bit different schedule. Pfizer actually, in their phase 3, sorry, this was an older slide, gave it to 16 and above, and then Moderna was 18 and above. These are huge trials, so together it's 74,000 participants, half got vaccine, 2 doses. And so Moderna, sorry about this. So Moderna will have its EUA evaluated by the FDA Advisory Committee on December 17th, and Pfizer had its EUA issued on December 18th. So both of these are now in the process of becoming available under emergency use authorization. It's been approved for Pfizer, and it's hoped that by the end of this week, the same will be true for Moderna. There are 2 viral vector, these are non-replicating viruses. One is a chimpanzee adeno, and the other is a adeno 26. virus that does not cause disease, and it's non-replicating. And you've probably heard about these vaccines that are in trials. Janssen's is 1 dose. I won't go into this because I know that somebody else is going to talk about these, but both of these trials are ongoing in both national and international sites. And then there are 2 protein subunit vaccines. So one is made by Novavax, and this is a nanoparticle vaccine with an adjuvant, a proprietary adjuvant called M1, 2 doses, and it was supposed to start in October, and every day we are just waiting for the FDA to sign off, so maybe tomorrow. And then the Sanofi vaccine had a little bit of disappointing news with their phase 1 trial, and so they're having a delay, and they expect to start in January. The ones that are circled are the ones that we've been involved in. So now I'll really turn my attention to the messenger RNA approach. So this is a diagram that just shows that you take the messenger RNA, which is made in a laboratory from plasmid DNA. It does not contain any infectious particles. None of these vaccines do. And it's just the code for spike protein. And it's the code for the stabilized pre-fusion form of the spike protein. And it's injected. And I think I'm not 100% clear on this. I have to be perfectly honest. I think that it's taken up by the antigen presenting cells, whether how much it enters the muscle cells or it's just injected into the muscles and the antibody presenting cells pick it up. That's something that I'm not entirely clear about. But anyway, you put the messenger RNA into the body, it enters the cells, and then it has the cells make spike protein through a process that's called translation. So this is a little, just a schematic, a cartoon of how these messenger RNA vaccines work. This SAM is for self-amplifying messenger RNA, and they include, the messenger RNA is able to produce the replicase machinery necessary for self-amplification of the messenger RNA. So you can see that it can make more protein. And this platform is not being used right now in either Pfizer or Moderna. So I just mentioned it because it's on this diagram. So this is the replicating messenger RNA, and this is what the 2 companies are using now. And so they inject the messenger RNA, and it's in this lipid nanoparticle, and that lipid nanoparticle allows it to bind to the receptor and enter the cell in a little vesicle that it kind of steals from the cell membrane. So it covers itself, the lipid, the LNP, also protects the messenger RNA from getting attacked by enzymes that could degrade it. So it also lets it live a little bit longer. So it comes into the cell, but it doesn't go into the nucleus. It stays in the cytoplasm, and then it uses the ribosomes to make amino acids, which are then formed into spike proteins. And then the spike proteins are expressed on the surface of the cell. And so here they are. So you now you're using our cell's machinery to make these spike proteins. And you don't need a manufacturing plant to do it. So it's a very smart way to make vaccines, and it has a lot of advantages. And so once the protein is on the surface, then it's detected by our immune system, and we make the type of responses based on the lipid nanoparticles and the messenger RNA. It really drives the type of immune response that we talked about earlier that we'd like to see. And so then you're immunized. And then the hope is that if you're exposed to coronavirus, if you remember that 3D picture, then the spike protein will be coated by our antibodies and it won't be able to get into our cells. So what are the advantages of messenger RNA as a platform? The other vaccine manufacturing platforms that we've been using for years that took decades to get vaccines into people were fixed facilities, and those facilities tended to be very specific for the production method and the product, and it was costly and inflexible. If you needed to change your product in any way as you learn more about it, oftentimes you had to renovate or make new facilities. And there are many complex process steps, and that adds very much to the cost and the time for ramping up and scaling up vaccine manufacture. And the messenger RNA-based vaccines really have a lot of advantages over that. As I said, they use the cell's natural physiology to make spike proteins that make really designer immune responses, we're hoping. They're uniquely suited for rapid design and development and scale up because you just make the messenger RNA for that precise protein that you want. And so there aren't other antigens in there that you have to purify your messenger RNA. You just make what you want, and that's pretty much what you get. It doesn't enter the cell nucleus. I'm asked all the time if this can get into your DNA and stay with you, and it gets degraded in a number of days and it never gets into the nucleus, as far as anybody knows, to interact with the genome. It's non-replicating, so it has its own stop and start. You know, some of the vaccines like measles vaccine and polio, they're live vaccines, and so they can be transmitted sometimes, and they have issues related to that. What are the disadvantages? Well, this is all brand new, they do cause inflammatory responses. That's why we get some reactions that we'll talk about, and that's why we get immune responses as well. And so we're just watching very carefully to see what the inflammatory responses are like, and to see if they're self-limited, and to see if there are any residual effects. And I'll talk a little bit about those in a few minutes. And we don't know very much about the durability and long-term safety because we're moving very quickly. So now I want to turn to the clinical trials that we now, with messenger RNA, and as I said, Pfizer and Moderna now have results that are available for us to scrutinize. So this is the publication that came out on the same day that the advisory committee met with the FDA. And then the next day, the emergency use authorization was granted. So it was published in the New England Journal. And I don't know, I'm not a sports person, but I know that's Yogi Bear. And I think that they really hit the ball out of the park with this vaccine. It was just much better than anybody expected. Here's the just the overall demography, just to have a look. It is mostly a white population. Pfizer did try to add another 14,000 subjects on the end of their trial to try to make it a more diverse population, and this is what they ended up with. And they had a good representation of elderly and people with some of the comorbidities. So the overall efficacy was 95% with pretty tight confidence limits. And then, so that's the primary endpoint. These are all secondary endpoints, the subgroup analyses. But what's pretty clear is that for the most part, you see efficacy across the board, regardless of race, ethnicity, age, gender. You know, the confidence limits for some of these are pretty good, and for others they're wider, and we would hope that we'll be able to get more information, but I think so far so good. These are the reactions, and so what this shows, this is vaccine and placebo, and these are the reactions. So the first is pain at the injection site, redness, and swelling. And it's the same thing here. These are the 16 to 55-year-olds and the greater than 55-year-olds. And the top row is dose 1 and the bottom row is dose 2. And so what you can see is that pain is the major finding for local reactions, and it's present in most people. You see it more in younger than older adults, and kind of surprisingly, because for other vaccines this isn't true, it doesn't increase with the second dose. It's about the same. And then you can't see on this, but most resolve in 1 to 2 days, and the most severe you can see is very small. I think it's, you know, less than 1 or 2%. These are the systemic events, and so this is, if you can't read it, I'll just read. The x-axis is fever, fatigue, headache, chills, vomiting, diarrhea, muscle pain, joint pain, and use of antipyretic medications. And so you can see that the most common are probably fatigue and headache, and that most are mild, but there are moderate and severes. Again, you see it more in the younger groups than in the older groups, and most resolve again within 2 days. It's more pronounced after dose 2. This is dose 2, and this is dose 2, compared to dose 1 for systemic events. And severe symptoms were uncommon, except for fatigue, which was in 3.8%, and headache in 2% after dose 2. Fever after dose 2 was seen in 16% of younger and 11% of the older group, and a lot of them elected to take antipyretic and pain medications because they were uncomfortable. This is vaccine efficacy against COVID-19 greater than at least 7 days after the second dose. And so this is just what we saw before, that the efficacy is about 95%. But the other thing that is worth noting is that we could take a little glimpse at what the effect of a single dose was, and the efficacy observed between 7 days after dose 1 and dose 2 was 52%. And in the first 7 days after dose 2, it was 91%. And then it reached full efficacy after that. And so this is really reassuring that there's a pretty rapid immune response that kicks in. You know, it's partial, it's not complete, but it kicks in after the first dose, and then quickly there's an anamnestic protective response after dose 2. And this is what the survival analysis looks like, and it just reiterates what I just said, that there's an early onset of partial protection. And so what you see is at about 12 days, that's when the placebo recipients and the vaccine recipients go their own way. And you can see here that's on or maybe even a little bit before 12 days. And so it does kick in pretty quickly, and that's nice. And this just shows the solid dots are the severe cases, and you can see here that but one, I think they had 10 and 9 were in the placebo recipients. Okay, so I wanted to give Moderna its due, but I do have to say that I'll go through it a little bit quickly because the results are so similar. So I don't know how many of you know who this guy is. I looked up, and he is Giancarlo Stanton, who I guess is the leading home run hitter for the Marlins, although I don't think he's with the Marlins anymore. So this is here to say there was another home run. Moderna also had to change its strategy toward the end of the study because they had a deficiency of minority populations, and Operation Warp Speed made them slow up getting to their 30,000 volunteer goal and just recruit minorities to try to get a more diverse population. And so that was done, and there's about 20% or so that are people of color. This was just a US study. About 25% were over 65. They had a different definition of elderly. So these data are not published, but they're publicly available in their VRBPAC packet that will be reviewed on December 17th. And they have lots of addenda. So I did my best to kind of just go through and try to pick out some highlights for you to see. Basically, this is the primary endpoint is prevention of COVID, and the efficacy was about 94%, again with pretty tight confidence limits. And this is with 196 people who were endpoints. And so this is actually a final analysis because the goal, it was an event, all these trials are event driven. And that's why they're so large. All of the trials are about 20 to 30, even higher, 1,000 people, because the hope was to be able to get to the goal, and most of them, the goal is somewhere around 150 or so endpoints to get to that goal more quickly so we can get a vaccine more quickly. And what happened was that we got into the third wave, and it was like, weeks, not very many weeks, between when they hit the first interim analysis point, which was, I think, 90 something, and their final analysis point, which was 150 something, and they bypassed it before their cutoff. So they got the efficacy results very quickly, and the efficacy is similar in the different age groups because the confidence limits overlap, but the study's not designed, the primary outcome is all participants. So that's a subgroup analysis, and we'll need to get more information on that. And these are mostly, I believe, ambulatory elderly people. And I wonder in my own mind how well they represent elderly who are in long-term care facilities. So the other results in Moderna are very similar, and I decided not to go through them with you. So the efficacy was similar across subgroups of age, race, ethnicity, underlying morbid conditions. They saw a cutoff between when the two curves diverged in the survival analysis at about 14 days. And so they saw a similar observation that there was efficacy after the first dose, and that there was early efficacy after the second dose. So it looks like it behaves in the same way. They had, I think, 30 severe cases, and all of those were in the placebo group. So these were really beautiful results. So let me just check on the time here. I'm getting near the end, so I want to wrap it up. So what do we know and what don't we know? We know that COVID illness can be prevented by vaccination, and the spike protein seems to do the trick. There is a favorable safety profile, but the reactions aren't negligible, and that will take public messaging so people expect it. They might feel a little bit bad, but they won't get COVID. It shows that mRNA vaccines are a promising approach and a fast approach. We got from genetic sequence to efficacy and an EUA in 11 months. What don't we know? We only have 2 to 3.5 months of safety. We don't know the duration of protection. Now, once we have the EUA, the trial participants will likely be offered vaccine if they receive placebo, and encouraged to stay in the trial so that they can be observed for safety. And you know, there are ways that people are looking at to see whether there's evidence of waning immunity or enhanced disease in vaccine recipients. So we're trying to keep them in the trial, but everybody will be vaccinated, and some will have to break out of the trial if they can't get the study vaccine in time. But we're hoping the companies are going to roll out these vaccines, as Pfizer is doing now, very quickly, and try to keep people in the trial getting the same vaccines. We don't know if vaccine prevents asymptomatic infection or transmission, and we don't have a correlate of protection for future trials, which means that bridging studies will be more difficult, and protection so high, it will be hard to get a correlate of protection. Okay. So I wanted to just give you a couple of shots about what we did at our sites. So we worked very hard to have a diverse community and worked with community leaders. This is the president of University of Maryland, Baltimore County. This is the president of our university. We opened up a testing site in a Latinx community at a nonprofit organization. This is where the implementation plan under the EAU stands, and you're all probably aware of that. But for the new trials, we know that healthcare workers and long-term care facility residents are going to get vaccines, and the 2 protein adjuvanted vaccines are still placebo controlled, at least to start with. And so we'll have to see how quickly these other phases of implementation of the EUA work, and this is the planned timeline for that. This is just a look at how the public feels about accepting vaccines, and there's a lot of vaccine hesitancy, which is something else that we have to deal with. So in the future, we're going to have to work with community engagement. There are plans to do studies in pregnant women and children, and we'll need to look at long-term safety and hope we can get a glimpse at efficacy. Thanks. So I'm happy to take any questions. I don't know if we have what we have time for, Glenn.
Yeah, just got time for a couple. questions. If you go to your question link, the ones that are published, you'll see the questions waiting for you.
Okay.
Or I can read it.
Okay, why don't you, that might be easier, just to make sure I can do this okay.
Is it fair to state that the non-serious adverse events mRNA vaccines are more common compared to other vaccines, for example, influenza vaccines?
I do think that that is my impression. Obviously there hasn't been a direct comparison. A lot of people like to compare these vaccines to Shingrix, and there seems to be a similar reactogenicity profile with worsening reactions after the second dose that you see with the zoster vaccine Shingrix.
One person comments that at my hospital, we are trying to figure out how to deal with sick days after the first and second shot in healthcare workers. Any thoughts about approaches to management of the side effects?
So I think that if you develop fever, you'll have to follow your hospital's policy. I would hope that they would be understanding if they're able to be. And then, for your own well-being, most people are not comfortable pre-medicating with anti-inflammatories or antipyretics, but certainly you can take them for symptomatic control. It is an issue that needs to be addressed, and it's complicated.
I've heard that COVID has already mutated with East and West Coast versions, and I might add also Great Britain versions and a variety of other areas. If this is true, will the vaccine be effective for multiple types, or will we be chasing its newest version every year like the flu?
It doesn't seem to have the same variability as the flu. We don't know what the durability of the protection will be yet, so I can't answer that. But from the virus mutating, so far we don't have evidence that it'll mutate itself out of the current vaccine, but we'll have to wait and see, and that's being studied.
Okay, hang on a second. Now I'm having trouble moving this stuff, but for the non-replicating mRNA vaccines, is the mRNA molecule only translated once so that one mRNA copy equals one spike protein molecule?
That's a really good question. I don't know. Do you know?
I don't know either. It is an excellent question. We will need to defer and get back with appropriate expertise. Do you predict that widespread use of the SARS-CoV-2 vaccines will shift the infectious population from symptomatic to mostly asymptomatic so that it will be difficult to follow transmission events unless more asymptomatic people are tested?
So I don't believe that is going to happen. I just don't think we have any evidence right now that that's going to happen, but we don't know what impact the vaccine is going to have on viral load and transmissibility. There are some studies that are being set up to be able to address those issues. So it's a possibility, but I think actually it's probably unlikely.
There was one comment that the Sanofi protein vaccine is being delayed because it didn't work well on the elderly, but it would be ideal for pregnant and immunocompromised. It's too bad they are holding back for the larger population. Do you have any thoughts or comments on where things are going with this particular vaccine?
Well, so don't forget Novavax. So Novavax, that's the one where every day we think we're going to start tomorrow, but I think we're just about there. And so that's an adjuvanted protein vaccine. And that could also be used for that population. ACIP did debate
Give Karen, give her a second. Go ahead.
Oh, okay. Did my,
We lost you.
okay.
Useful freeze.
So there are plans to do trials in children and then in pregnant women. There is a little bit of safety data just because of inadvertent pregnancies, and there were no signals. There's no a priori reason to think that the mRNA vaccine would be harmful in pregnant women. It makes the spike protein. I mean, it kind of does the same thing. But right now it's not recommended for pregnant women, but trials are proceeding. It's supposed to be first in children and then in pregnant women. I think Sanofi, the little bit of insight that I had is that they had some purity issues, and I think they need to get cleared up before they can really go ahead. So I'm not sure if it's just the age thing. I think it's also a manufacturing glitch that needs to get fixed.
Okay, I think we are about, actually, one more question here. I have heard in the media, social media, the vaccine will not prevent the spread of the virus. Is social distancing still required?
So it's that we don't know. It could be perfectly good at preventing the spread of the virus. We have no idea. And as long as we don't know that, we need to social distance and wear masks to protect the people around us, even if we're vaccinated.
Okay, I think we're past the 5 o'clock hour, so I think at this point we'll probably go ahead and stop the questions. Karen, we've actually gotten a number of nice comments commenting on the fact that people really appreciated the talk. Really appreciate your taking your time to give this presentation. And I would note that this will be available online through our webpage for those who are interested in it or who may want to tell other people about it. It's, I think, covered an amazing amount of material. And again, my sincere appreciation for your for putting this together and taking the time to be with us.
Sure. Well, thanks for having me. I appreciate it. Everybody take care.
Take care, everyone. And I think we will close out the session for today and stay safe.
Okay, you too. Bye-bye.